Well, I hope everybody's having a calm day. This is my first attempt at trying to put an audio track to our PowerPoints. I'm going to try to see if I can do this section by section. So this is, again, the first attempt at this section. It's the small intestines in chapter 17. So at this point, we've looked at the digestive system from the oral cavity through the esophagus, stomach. Now we're in the small intestines. Again, the small intestines is not named because of the length. It's named for the diameter. It is the smaller diameter too, but by far the larger or the longest portion of the whole alimentary canal. It receives chyme from the stomach and the liver and the pancreas secretions, what we had just looked at. One of the last things we looked at while we were together in class, that the liver produces bile. That's what the liver does many things. We looked at the liver producing bile and the bile being stored in the gallbladder, and then how it joins the bile, joins with the pancreatic duct for the pancreatic secretions. Now the small intestines is going to be the end result of the process that we talked about with digestion. The large intestines doesn't really have any breakdown. This is where it does. So this is where the digestion is complete and the nutrients of the chyme, as we called it, as it was leaving this mixing with the gastric juices, as it starts breaking down, then it's going to be absorbed here. Now, what is left over is going to be transmitted or transported into the large intestines. And at this point, really, it is where we're pulling out the nutrients, all the stuff that the nutrients of what we eat and ingest is going to be, the good is going to be pulled out and the bad is going to be thrown out. So, now the small intestines is made up of three parts: the duodenum, the jejunum, the ileum. You need to remember these in order. The duodenum is the start of the small intestines. It is where the pancreas and the liver and the gallbladder dump their secretions. As it says here, it's the shortest and most fixed portion of the small intestines. That's because this is stuff that's just coming out of the stomach. The jejunum, the middle portion, is thicker and more active than the ileum. This is where a lot of that, we talked about segmentation, how the small intestines, how the GI tract will constrict on either side of the food substance, and then it'll kind of massage it or kind of mix it up. And we call that segmentation. And it was referred to as a mixing movement. The jejunum is the most active part for that. And the last portion is the ileum. It is what's going to connect to the large intestines. It also has specialized little lymph nodes that we'll look at called prior patches. They are the things that really help protect us. It is amazing how nasty the world is and how much is really trying to get into our system. I mean, we still need lymph nodes, things protecting us all the way at the very end of the small intestines. This is past the stomach and that very acidic gastric juice, the hydrochloric acid, and all the breakdowns, still having to fight off infections. The jejunum and the ileum are suspended by the double fold of the peritoneum called the mesentery. We looked at that. It transports blood and lymphatic vessels and nerves to the wall of the intestines. I talked about that in lecture and how you'll really see that when you get into lab. That is one of the more interesting things to see in dissection, which, again, I don't know what's going to go on with that. But anyway. Double fold of peritoneum called the greater omentum drapes down from the stomach over the large intestines and folds of small intestines. The picture is coming up. In here, we don't get into what they do. And as a matter of fact, there's a lot of confusion about what the omentum does, the greater omentum. This is the picture that's supposed to show this, and it's the mesentery here. It's hard to draw something that's clear, but you'll see that it's going to be this clear, very tough membrane, and you can clearly see the arteries and veins and lymphatic tissue and even nervous tissue coming through. Now, again, the picture here in the middle is supposed to show the mesentery, how it really is this, there's adipose tissue in this long extension of the peritoneal cavity. You can see the difference between the two membranes. On this picture, you can really see what the greater omentum looks like. Again, lack of a better way to say it or call it, I call it a fat curtain. It is just a layer of adipose tissue there, and it has, again, some odd things that people wonder about what it does. It does have the ability, as far as I remember when we studied it, to move slightly. If you have an infection, it can kind of move inside the abdominal cavity to try to give protection, what it appears. Now, this section, again, understand that when we're talking about the small intestine, since it is the area of the absorption, this is where we're going to have a lot of surface area. There's going to be a lot of things trying to increase surface area here. So we'll have these finger-like projections of the mucosa into the inside of the cavity of the small intestines, inside the lumen, I should say, if I want to be more correct. And this is going to cause a huge increase in the surface area and helps for absorption of the digestive products. Now, the interesting thing about this is they're lined with simple columnar epithelium, and then underneath it there's going to be connective tissue with blood vessels. They're going to be absorbing the nutrients, all except one type. One organic molecule cannot be absorbed through the blood vessels, and so we need a lymphatic capillary, a specialized one that we looked at before in the lymphatic system called a lacteal. The lacteal is designed to absorb the dietary lipids as we break them down and absorb them through our small intestines. Now, these villi with their simple columnar epithelium, we'll look at slightly on this next part because it talks about these microvilli. So if you can imagine that each one of these cells has these little waves in the surface that's in contact with the chyme, you'll see that all along the surface there are little villi, so they're called microvilli. And all this is doing is really trying to increase that surface area. Again, increase surface area, increase absorption rate. Now we'll have special type of intestinal glands. We're not going to, refer to them as the crypts of Leberkuchen. Again, I shouldn't say that. It's kind of joking, but I don't know how to pronounce that. But I do know, these are just specialized glands that are going to help with the secretions, and I'm not going to hold you accountable for those right now. But what I do want to talk about, and these are kind of hard to explain. I feel odd with my hands tied where I can't draw or try to describe this. And again, I don't know how to make these videos, so this is a first attempt. But these Plicae circularis, or circular folds of the mucosa, which also increase surface area. So they're going to be these rings, these ridges that are rings that go around the inside, and off of the rings the villi stick out. I believe the next slide will have a picture of that. I hope you can see it. Well, I was wrong. This is, sorry, I was wrong. This is a picture of a villi. And so it looks almost like a little thumb, like, hey. But anyway, inside of the villi, you can see the capillary bed with this lacteal in the middle of it. And those lacteals you can kind of see are a little bit larger than the capillary bed that's around it. And then you can see the little simple columnar epithelium that line it. I believe the next one you will see is going to show the Plicae circularis. And again, I am wrong. This is the simple columnar cells, and so this is showing the microvilli on the top. the microscopic actual picture on the right-hand side, you can see them. Again, that picture almost looks to me, if you organize that a little bit more, as far as the organelles, it could look like Bart Simpson there. But anyway, this is just trying to give you an idea of just how much surface area is being increased. If we were to look at that and lay it out, that would be exponentially larger if I was just looking at the surface area than what the top of that one cell would be. And now we finally get to the Plicae circularis. And so hopefully you can kind of see that this is what we're talking about here. That if you looked at that cross section at top on A, you would think, and just looking at that, you would think that the little ridges that stick out were the villi, but it's not the case. Those things that we'd see when we cut it across are actually cross sections of those circles that go around it. But all along those circles would be villi, and then on the top of the villi would be microvilli. I hope that makes sense. It is really a great mechanism because, again, if you can imagine that where that, and the picture A, if you can imagine where there are little contractions on either side of a little segment of fluid in there. And remember, the fluid that is in the small intestines is going to be very watery. And so they would take it and it would slosh it back and forth, and it's forcing all of that liquid to run through those microvilli in the villi. So hopefully you can kind of see that. And all of that is adding to this exponential surface area and efficiency of absorption. Now, this is talking about some of the secretions of the small intestines. Again, one of the big things, there's going to be a lot of alkaline mucus that's going to be released here all throughout, but in particular the duodenum, but all throughout. We are dealing with something that is much more acidic than the environment that's around it. And so there's going to be these there's going to be a lot of alkaline mucus. It's Brunner's glands that are ones that are going to secrete this, that help protect it. And again, certain stimuli, if something is a little more acidic than others, it will be active. Now, there's a constant watery fluid. These intestinal glands secrete a watery fluid, and this is what helps the digestive enzymes. As it says here, it doesn't have them in it, but this watery fluid is what provides the medium. Again, everything is about water. Anytime you have any building up of molecules, any anabolic reaction requires water to be removed, and so it's called dehydration synthesis. I'm synthesizing or building something by removing water. If I am breaking something down, so I've got something large, which is what we're trying to do here with digestion. We've got these large molecules, these large carbohydrates, and we need to start breaking them down. So I need to have water that gets in the middle of some of those bonds, and we called it dehydration synthesis, right? I'm going to break something down, or I'm sorry, I just said dehydration synthesis. I meant hydrolysis. Hydrolysis, I'm going to break something down, lyse it with water. Hydrolysis, using water to break something down. And that's the process of catabolism, which is what digestion is, breaking things down. And so while these intestinal glands and their watery fluid don't have any enzymes in them, they are used to help maximize the enzymes. Remember, if we went back to talking about enzymes, the enzymes were kind of the comfy couch. They let somebody sit, they let this molecule sit on it in just a way that it exposes that little area for water to go in and cleave them apart. And so you need both water and the enzyme to be effective. And so then the enzymes, which are going to be released, they're going to be released by the simple columnar cells. And what happens is A lot of books, you might hear this later on, will call them brush border cells, because if you go back and look at the top of that little picture that I said looked like Bart Simpson, it looks like a brush. So they called them brush border cells. And that, and here it says the enzymes and the membranes of the microvilli, but it is the brush border cells. They are producing it, and they're caught. They're going to be all around those microvilli. So when the water's swooshing through them with all the material, then we've got the final breakdown. Remember, we talked about when we're breaking things down, if I have a carbohydrates, a complex carbohydrate, there's hundreds, if not thousands, of simple sugars, got tongue-tied there, monocarbohydrates, that are bound together in this big thing. So think of it like a brick wall, and each brick is a monosaccharide, a simple sugar. And the wall is what we eat, and I've got to continually break it down until we get to the little bricks. And so this middle layer, sucrase, maltase, and lactase, these are the ones that break down the main disaccharides. Usually everything will get broken down into finally where there's just these two sugars together, these disaccharides. And the three main ones are sucrose, maltose, lactose. And so if we don't have, let's say, lactase, which is the enzyme to break lactose, we cannot break it down and absorb it. We become lactose intolerant. And so sucrase works on sucrose, maltase works on maltose, lactase works on lactose. And they break down. I cannot absorb, those little brush border cells cannot absorb the monosaccharides until they can't absorb the disaccharides until they're broken down to monosaccharides. I hope I haven't messed this slide up. But anyway, I'm trying. So peptidase, peptide bonds are what hold together the amino acids. So when proteins finally are getting broken down into small chains of amino acids, we call them peptides. The peptide bonds are what connect them together. So peptidase is the end breakdown. So now we need to get these long chains of amino acids broken down into individual amino acids so they can be absorbed. And then finally lipase, which is another one that's going to break down fats, try to take those fatty acids off of the glycerol. So this is the list of the major digestive enzymes. I will tell you when I'm looking through this, pretty much anyone that doesn't end in ASE, and now nucleases, that one at the bottom of the pancreatic enzymes, understand it does end in ASE. They just put an S at the end of it because there's no one, it's a catch-all, so it's plural. But anyone that doesn't end in ASE is all about proteins. The other ones are either going to be a carbohydrate or a lipid or a nucleic acid. There's only one that breaks down nucleic acids, and that is coming from the pancreas. We do have a lipase coming from the stomach called gastric lipase. We also have a lipase coming from the pancreas called the pancreatic lipase. A lot of these aren't as complicated as they sound. Now there is one carbohydrate enzyme that does have ASE, but it starts with carbo. Hopefully that's a big keyword in it. And it also has a peptide, which should clue you into peptides. So carboxypeptidase is one of the ones that helps really break down things into smaller amino acids. All right. And then the peptidase here are the intestinal enzymes. This is tough, I know, and I see that the one on the bottom there. But as a general rule, very few of these enzymes do not end in ASE. If they do not end in ASE, automatically you should think proteins. There are a few that do end in ASE that work on proteins, but they're fairly well named. Now, the regulation of small intestinal secretions, the regulations are going to occur in a couple different methods. So the mucus secretion is going to occur in response to mechanical stimulation. So as the small intestines will start the peristalsis, as it's moving the chyme along, as things get stretched or get moved, they will secrete mucus, because that's going to help things move along, but it'll also help protect the wall of the small intestines. Chyme is going to stimulate goblet cells to do the same thing. And it will also stimulate the secretion of that water in there to help kind of use the enzymes. When the distension of the intestinal wall, the nerve plexus, so we talked about those submucosal and the mesenteric plexuses. So as the intestinal wall stretches or moves, it's going to stimulate. We'll have a reflex in that. That's going to have a parasympathetic reflex that's going to trigger the release of different enzymes. So there's all kinds of fail-safes as it's moving through. Some of it's going to be just the feeling of things, whether it's like it says at the very top, the irritants of gastric juice, or just the stretching of knowing something is moving is going to trigger certain things. It is an impressive thing that a lot of us don't think of, and whenever we do, we just think of the I hate to say it this way, but kind of the gross aspects of it. But it is an amazing system. So this is the absorption portion of it. I didn't realize it would take this long, so I'm going to keep this going. I don't know if this is going to help anyone or not, but this is my first attempt, like I said, but I'm going to keep going. So in this, we end up kind of talking about the breakdown of these carbohydrates. So villi, increased surface area for absorption. Again, the small intestine is the most important absorbing organ of the whole alimentary canal, the whole GI tract. This is the star of the show, really. It is so effective, as it says here. Even if as we get to the ileum doesn't do as much, simply because there's not as much to do. Most of anything useful has been taken out by the jejunum. So when we look at carbohydrate digestion and absorption, the salivary and pancreatic amylase help break down starch and glycogen into disaccharides. Again, starch is plant stored glucose, glycogen is animal stored glucose. And we want to break it down eventually into monosaccharides. But right at this point, they are huge molecules of hundreds and hundreds of packed glucose molecules. So we start breaking those down. That is why it's so important to chew your food, because the mechanical breakdown helps this. The intestinal enzymes break down disaccharides into monosaccharides, and then the monosaccharides are absorbed by way of facilitated diffusion and active transport into the blood vessels in the villi. Facilitated diffusion, remember, that means that there are built-in channels that help absorb or move something from one side of the membrane to the other, but it does require energy. There's a give and take on this. We're not going to get too far into that, but that is how it works. Protein digestion and absorption. So pepsin in the stomach starts breaking down proteins into polypeptides. Now remember, protein is just a name for a specialized molecule. And at this stage, when it's a protein, it is not only just a long strand of amino acids, but they'll have this really coiled, twisted, and turned and very unique shape. And the first thing that has to happen is that shape has to unravel. It has to kind of fold out, and then it becomes this long chain now that we commonly think of, and that's a polypeptide. So pepsin starts breaking down the proteins into these larger, less structured shapes of chains of amino acids. And then the pancreatic proteinase, both trypsin and chymotrypsin, start breaking down proteins into smaller proteins, which we'll end up calling peptides. Peptides are simply when it's only a couple of dozen amino acids that are attached together. So then by the time it's gone through that, now we're getting into certain areas where, I need to break it down to amino acids to absorb. So finally I have intestinal peptidases, which are there to break them down into the amino acids. And finally the amino acids are absorbed by active transport and into the blood vessels of the villi. And I must correct something here on the last one. Facilitated means that the body makes it happen. The carbohydrates happen without any give and take. That's what facilitated means. There's no energy. We're going to make it happen. In this one, it is active transport, where it does mean there is a cost. I misspoke on the last slide, and I apologize. Now we get to fat digestion and absorption. This is kind of interesting. Fats are emulsified by bile salts. We talked about this in class. The emulsification process is where these molecules that have both a hydrophilic and hydrophobic portion, they kind of get around and they help separate the oil, the lipids, into smaller droplets and smaller droplets. And then we can start using the specialized enzymes. So fats are digested mainly by enzymes from the pancreas and the small intestines. There's some in the stomach, but it's tough because of the acidic environment. So fats are digested into their two main parts. There's a glycerol backbone and then 3 fatty acids. And so that's what the breakdown is. Digestion, again, means to be broken down. Fatty acids and glycerol are absorbed by the process involving several steps. And as they start breaking them down, they get pulled across, but they don't go into the capillary beds like the carbohydrates and proteins do. They have to go into the lacteals. And it's because lacteals in the capillary beds, there is a diffusion as it goes across this simple squamous lining. But these molecules are too big to have that happen. If you remember, in a lymphatic capillary, we had those little trap doors that were used to sense the pressure. And so those trap doors actually allow larger molecules in, which is how they deal with pathogens. Sometimes pathogens are actually a fairly large molecule on the molecular scale. And so those trap doors allow things that are a little bit larger in, and that's why the lipids are used that way. This is a picture that kind of shows. It is kind of sad. We don't get into a lot of this. In order to go from the intestinal tract on the inside through the cells that line it into the lacteal, there is a pretty big process. As the fatty acids are broken down, they will come in contact with the brush border. They'll be taken in. Fats, again, can move through cell membranes like they're not there. Remember, that is the one advantage of them. So as they get close, these things will move through. Then they will go through the endoplasmic reticulum and be packaged into things called chylomicrons, which is just a word for a fancy word for like a very small package, basically. And it's going to go through and then get into the lacteal. Now, on here, it goes through some of the things that are how they're absorbed in the intestinal tract. This is not going to be a big part of this. We don't go into this really in depth. Monosaccharides, simple sugars, go through both facilitated and active transport, but you do need to know they go into the capillaries. Amino acids, which are from proteins, you need to know that, are absorbed by active transport. I'm not going to hold that accountable, but you do need to know those go into the blood capillaries. Fatty acids and glycerol. Now, these are going to diffuse and facilitated diffusion is just going to be what helps the glycerol. Simple diffusion is what lets the fatty acids into that. As we said, they get changed into chylomicrons. But what I want you to know, first and foremost, is that the fatty acids are going to be absorbed by the lacteals. Now, again, on the bottom part here where you see B, There are very few that will be also absorbed into the capillaries, but that is not normal. What I want you to associate normal with on this, when I say normal, that is normal. I'm saying that that isn't the main way. What I want you to associate it with the main way is that it goes into the lacteals. Electrolytes in water, I'm not really worried about. Electrolytes are going to be all of those minerals, sodium, potassium, calcium, all of those that are coming out. They're going to go straight into the blood capillaries, and so, as well as water, and water is just by osmosis. So again, the small intestines is going to have two types of movements: peristalsis, which is simply moving the chyme forward, and then segmentation, which is a little bit more complicated. We talked about it's where these little contractions on either side of that chyme go, and then they shake it up back and forth, basically. Always remember the parasympathetic is stimulatory to both of these, where the sympathetic is inhibitory. This is one of the big things, that the sympathetic, the fight or flight, the main thing it's trying to do is it shuts down the digestive system, because at that point we're not worried about absorbing things. At that point we are worried about using our reserves, fight or flight. I'm going to stand my ground or I'm going to run. Either way, I'm going to have to burn energy at a great fast rate. And so I'm not worried about absorbing it. Where parasympathetic's just the opposite. It's not worried about all of that stuff. It's worried about digesting things and absorbing them. Also remember, parasympathetic is mainly the vagus nerve, cranial nerve 10. Now, if you get a small intestine becomes overdistended or irritated, a strong peristaltic rush that may sweep contents quickly into the large intestine and result in diarrhea. Diarrhea we're going to talk about a little bit when we look at the large intestines, but understand diarrhea is one of those things that happens. It's not pleasant to talk about, but we will talk about it when we get into large intestines. Diarrhea is actually a very dangerous thing. Water is everything, and most of the water, you're surprised at how much water is going to be in the digestive tract, and then it gets reabsorbed. And while that might sound terrible, it is vital. If you have diarrhea, it is easy to become dehydrated and leach yourself of so many minerals because you're not getting the chance to absorb it. Now, there is a junction between the small intestines and the large intestines. It's called the ileocecal sphincter, and it's because the ileum of the small intestines is on one side and the cecum of the large intestines on the other side. And that is what is helping regulate the flow of chyme.